And something that really inspired me for the sort of the beginning, the first piece where I sort of like start the story is so like that's the water bottle that I actually used to sample water from the sea, and then the quote that sort of starts this piece is from an essay by Joan Didion, who is a writer, also sort of did most of her work in the 60s and 70s. She lived in LA and has really beautiful essays about sort of life in California, which I highly recommend for you guys. And so she has an essay called Holy Water that's really about the bureaucracy that brings water to LA. So she talks all about how like the water that is in your tap started its journey to Los Angeles like the day before. And there's people who have to decide how much water LA is going to need on any given day. And they like send the water out to LA. You know, it's a very interesting story that she sort of tells. So that's the beginning, is this idea of sources, water sources. Where do we get our fresh drinking water from? What is the source of water that goes into the sea? So like the original sort of accident, and now the how the water is maintaining the sea from drying up, because it's also in a desert. All the water that's going into the sea, so that's a sort of satellite image of some of these inlets, is draining off of the agriculture that's in the valley. So that sort of drainage, like from the irrigated agriculture, like, and then off of the agriculture goes back into the sea and sort of sustains the water there. But that also brings a lot of pollution and has increased the salinity really significantly. So the salt concentration in the sea has gone up over the past decades to a level that's more than what most fish can survive. So a lot of fish die-offs have happened starting I think in the 60s and 70s. So that's why the beaches are skeletons, is because the fish can't survive as the salt level goes up, and then so the fish will sort of die, and then the beach makes this really nice white beaches, which look good from far away. But the salinity too, there's a lot of work now being done to try and maintain it at what level it is, and sort of stop it from keeping going up. So they need to. Another interesting thing for me, interested in bureaucracy, obviously, is they have to redefine the word pollution. So like pollution from the agriculture is no longer now considered just like the water going in is necessary. So it's not polluting for water to go in, it's polluting for not enough water to go in. Because then if the water dries up, then it'll like create a lot of dust and then a lot of decrease the air quality. So pollution has been sort of like rearranged.
Rearranged.
Yeah.
Move that over here.
But yeah, that's the first. I'm talking way too long, I think. But that's the first one.
That's the idea. We want to hear you talk, and then you'll get questions.
Oh yeah, and you can interrupt me if you have questions before then, obviously. So stop me. But so then I wanted to sort of see. I don't know. Do you have a question now, or
no? I'm just moving away so they can see the next bit.
Sorry. Well, so then the idea was to sort of say, well, the water, has this high salt concentration, but it also has, the dead fish actually like provides a lot of nutrients that can sort of sustain this really interesting ecosystem. So the Salton Sea has some of the like most diverse population of birds in the state. A lot of it has like a really dynamic bird sanctuary sort of, because there's so little development in the wetlands that they can have like a sort of undisturbed habitat compared to a lot of the sort of Pacific wetlands that have been developed by humans. So I wanted to sort of show some of the like ecosystem through this microbiology. So I made a giant petri dish here in this like bucket where I have agar with nutrients, and then put just a little bit of the water from the water bottle onto the nutrients. So I think I made it so it was like 4% salt, about the same as the sea, and then it has some like yeast extract and sugar, sort of enough for the bacteria to sort of eat and grow. And then I was actually surprised by how much grew. I wasn't expecting there to be such a high density of things in there, but there was a ton of different bacteria that show a lot of the diversity that's there.
Is it continuously growing, or has it stopped?
It's stopped. Now it's really drying out.
To me,
So I think it kept growing and changing for the first like week or two, and then it sort of became static and started to die off after that. But so growing bacteria like this on solid media in a microbiology lab, only about 1% of all bacteria can grow in this way, which I think is really interesting. So this is only showing a very, very small snapshot of the diversity that's actually there. There's many more different kinds of strains that are present. But then sort of the sort of next step, I sort of approached this first as a microbiologist. I wasn't sure that this was going to be what I would be exhibiting. So I was like, okay, well now I want to, me move over or Victoria?
Sorry.
Now I wanted to sort of find individual species of bacteria from this big petri dish, isolate them and sort of understand their properties better, and try to identify what the bacteria was. And so the next stage is monoculture from ecosystems. So monoculture is something that is happening obviously in the agriculture around the sea. Each plot will have a single species of plants that the farmers are growing. And then in the petri dishes, I have a monoculture of bacteria, which is a single strain. Which I basically like looked on the plate on the giant petri dish for a colony that would be a single color. Like I was looking for things that were bright yellow, orange, pink, different things, and I would grab them and then streak them out on these plates to sort of isolate things out of the larger population, to show this kind of abstraction into monocultures.
It's really interesting to me as an artist listening to your process. And it sounds very much like an artist would work. Like you're looking and you're researching and then you're pulling stuff and seeing how it would work. And it's very similar.
Oh yeah, there's a lot of interesting connections there, I think.
I mean, in a way, it's like you were painting.
Sort of.
Really?
Yeah.
Because you were, in a sense, controlling the aesthetic by pulling out a particular bacteria, and you could kind of predict a little bit of where it was going to go, right?
Oh yeah, from the color that they were when they started,
That's cool.
That there's like a randomness to it, but then it's very deliberate from there to there.
Exactly.
Yeah.
All right. So
Okay,
continuing on.
So then, again, my microbiologist, I was like, okay, how, if these are bacteria that are sort of optimized or adapted to this high salt concentration, how high of a salt level can I get to? So I was interested in this idea of adaptation, like as the salinity may increase, or like what happens to the sea, like when there's gradual changes or more dramatic changes. The fish will die off because they can't adapt so quickly, but what happens to the bacteria? How much sort of leniency and room for adaptation is there already in the bacteria themselves? And so I had 3 of the different strains that I had collected from here. They're one on each sort of rack, and then across the rack is media with sort of nutrients to grow, and then salt concentrations increasing gradually from 0% to 10%. So 10% is really high when they're talking about extremophiles. Do you guys know what that word is? So like bacteria that are adapted to really extreme environments, whether it's high salt, high temperature, low temperature, they're called extremophiles, and they have a super interesting biology. But so 10% is when you're getting into like really strange territories, and you have to do some other sorts of special things to help them grow and help them maintain, which I wasn't adding any, I think you have to add extra magnesium, stuff like that, which I wasn't doing. But so from 0 to 10%, you can see there's a really dramatic change and a really dramatic difference in the different strains in where they can survive. So the top one can only survive in a very tight range, sort of between 2 and 4% maximum. So that one has a really sort of specialized tolerance to salt, which I think is really interesting, that I was able to find this sort of narrow range bacteria within this changing and very salty environment. And then the other ones are much broader. They have like an optimum. They've sort of sunk down, so you can't tell as good, but they used to be like a sort of curve. You could see the optimum, like the peak, and then they sort of go down. But they are very much more flexible until about like 8% salt, I think, is the top that I could get. But so that's adaptation.
Extremophile sounds like a good band name.
Yeah. That's good.
Did you see the Extremophiles?
Yeah, I think, well, philos in Greek means friend, so it's like friend of extreme environments.
Perfect. Perfect. So did your professor see this exhibition?
Yeah.
And what did she say? Did I meet her? I don't think I did.
I don't think you met her. I think you
I
didn't
think we
cross
missed each
paths.
other. Too
Yeah.
bad.
She was interested, so I had told her what I was doing, and
Yes.
then she was curious. She was like, well, I don't have time to think about this too much, but freeze the strains that you do find, because maybe there's something interesting in there. Because she's been working with salt-tolerant bacteria, especially and thinking about the ways that they may help plants grow in salty environments in particular. So she was really curious about, well, are there bacteria that are known to associate with plants in this set? And she pointed out one in particular. She's like, I think that this one, Polaribacter, that might be a plant-associated guy. So like maybe you should look into that one. And I was just so she was very curious.
And what was
about
her response to you actually doing this in a gallery setting?
I think she was really curious about it and really interested in the idea.
Yeah. But
so
I mean, when she came physically,
I think she liked it. Yeah.
You think she liked it?
Yeah. I mean, we didn't talk that much about it
because she's very strict research scientist, right? So to her, it's almost like an extracurricular event, right? That's
sort of, yeah.
Not necessarily that important. Which some scientists very much have that kind of attitude, and then there's others who we met who feel, and you, who feel like, no, it's actually not extracurricular. It's very much part of my process.
Yeah.
Can you speak to that a little bit, the difference and perhaps the importance, or does it matter?
Well, so Anne is really interested in, and she definitely recognizes the importance of public outreach and education in particular. Right. So that's really important to her. So I think that she definitely sees the artwork in that vein and more like a public engagement. I think she's also, often scientists are interested and very curious, but they don't have the time, I guess, to be able to really dig in and to sort of think about it. So I mean, the way that the art works for me in my own process is that it sort of takes me outside of the specifics, right, of the daily lab work and the focus of microbiology. It takes me outside and connects me to the okay, well, where does this water come from? Where is that going? The context. And so if we're talking about developing strains to put in agriculture to protect them from salty environments, salty soils, it's well, why is it getting saltier? Those are the questions I want to ask and I want to get at. In the end, and I want to think about what, if you do introduce a new organism, how's that going to change? And I think that's part of the science, and I think that you can explore that through many different ways.
I think it's so important because sciences up to now or recently have been just so reductionist. So you're absolutely removed from the social or wider or global context. And
well, you want to be removed, I guess, but you never are.
You never are, exactly.
I think it's the problem.
But the methodology that's taught is that you're going to do this bit of it, and anything else is a distraction, right? But in my view, I see more and more the importance of, well, you're doing this one atom, or this one molecule, or this one bacteria, and you're going to do this for the next few years. That's okay. That's good. It's not saying, oh, you shouldn't be doing that kind of very specialized, for lack of a better word, reductionist way of thinking. But if you zoom out, like you're saying, and you start looking at how that bacteria or that one molecule actually interacts with a larger picture, when you go back to your bench, it makes a difference.
Oh, yeah. It definitely changes the kinds of questions that you're asking.
Yeah.
Yeah.
So I think that's something that should be integrated into education. I don't know whose phone that is. Do you want to? It's your phone. Okay, don't worry. It's background music. So that's also something that everybody who's in sciences or that's pursuing science in this room should be aware of, and obviously you are because you're taking this class. But it's something that you should take Christina's lead on, because it doesn't in any way take away from your science. I think that's something that's a mistaken point of view. And a lot of artists, actually, people don't talk about it on the reverse.
Mm-hmm.
But I find that even with graduate students, sometimes I find it really hard to pull them in and say, okay, let's go to the lab and talk about and look really at what philosophically inspires you, or aesthetically about biology or physics, or quantum this or that.
Yeah.
Let's go see really. It's time consuming, of course.
Oh,
But it makes such a huge difference when they go back to the studio and start reworking the same.
Oh yeah.
It's hard to pull them in. It's not easy.
It's hard, especially I think because there is sort of this expectation that you do need to have the prerequisites, I guess. Like, and
The
that can be
education
really.
system is the problem, actually.
I mean, it's
It really
both the
is.
problem but also the solution, I guess, in a way. Like, it's challenging because there are a lot of things. Like, I think that's what you learn when you're a scientist and when you take science classes or when you're engaging with science, is that the more you learn, the more you realize how much you need to learn. And so that's sort of the challenge and the beauty of working with science as a scientist or as an artist or somewhere in between, that there is just so much there that it can be very daunting, I think, to sort of, and I think challenging to say, well, I don't need to do the whole thing.
Right.
I need to do just a little thing. I want to find like one thing that I'm interested and inspired by, and then it's sort of like a reductionist approach in a way, but not really. Like that you can narrow down, you can focus, and then you sort of try and make connections, like grab onto things like rungs of the ladder, sort of like as you're going along.
Yeah, exactly. And it also points to need to collaborate and to dialogue and move through different disciplines and just be curious, and you can go back to the little things. So having too many things going on, of course, is a problem too, right? You have to figure out where you're coming from. So
Yeah.
it's really amazing.